Bismillahirrahmanirrahim. What's up guys? My name is Yaul. I'm from GMM first 4th semester. So in today's presentation we will talk about the blood, its types, why the clinical correlation of the blood, and more the drugs affecting the blood, and the intrinsic and the extrinsic pathway of the coagulation of the blood if injury occurs. So in this case what we have to do with pathway follows the blood, and the thrombolytics and the folate cycle. So let's start it from the very beginning. We know that a normal adults have approximately 5 liters of blood. So in case of female, the quantity of this blood is a little bit lesser, but normal we have 5 liters of blood. Blood is a specialized fluid. It has 4 main important parts like the plasma, erythrocytes, leukocytes, and the thrombocytes. Blood has many different functions, including it helping transporting the oxygen and nutrients to the lungs and tissues. It also helping forming the blood clots to prevent excess blood loss, which we will study it later, and it also helping carrying the cells and antibodies that fight against infections. We will study also it later, like the intrinsic and extrinsic pathway, how coagulation process occur, bringing waste product to the kidney and liver, which filter and clean the blood, while it also help in regulating the body temperature. So in this table you can see clearly here, whenever the doctor gives you the CBC test, the complete blood count CBC is the short abbreviation of this. So here we can see the normal range of the white blood cell, the red blood cell, the erythrocyte, the platelets, the eosinophil, the lymphocyte, monocytes. So in case of leukocytes, we can see here 4 to 9.5. This is the normal range of the leukocytes. While in case of thrombocytes, while in case of erythrocytes, here you can see 3.9 to 4.70. This is the normal range of the erythrocytes. While in case of platelets, this may occur 180 to 320. And here also I want to mention the units. Here 10 power 9 it goes to the billion, while 10 power 12 it's a trillion. 10 power 9 against the million. So this is the normal unit of these range. So whenever here you can see the result, it's like a result of a patient. Here you can see in case of thrombocytes, erythrocytes, the normal range is 3.90 to 4.70, but in this patient have a 3.64, so which is not normal and less than the normal. So again this patient have a deficiency of erythrocytes. So by this table we can completely conclude or give idea about that we have a normal or abnormal blood range. So let's study the types of blood in more detail, like one by one. Number first is red blood cell. It is also called erythrocytes. We know that this blood contains no nucleus, so this is non-nucleated. The normal count of RBC is around about 7. In case of men, it is 4.7 to 6.1 million RBC in case of men, while in case of women it's a little less than the male, like at range around about 7.2 to 5.4 million RBC per microliter of the blood. So in case of children, it also have less than women, like 4 to 5.5 million per liter in case of children. So red blood cells are also called erythrocytes. The cellular components of blood, millions of which in the circulation of vertebrates give the blood its characteristic color and carry oxygen from the lungs to the tissues. This is the main important function of the red blood cells, to carrying oxygen, carrying oxygen from the lungs to the tissue. It is covered with a membrane composed of the lipids and proteins. So as I already told you that it lacks a nucleus and contains hemoglobin. Hemoglobin is actually a red iron rich protein that bends the oxygen. So the purpose of carrying the oxygen from lungs to the tissue is due to the credit goes to the hemoglobin. So now we will study the structure of the hemoglobin in detail. Hemoglobin is generally a blood carries oxygen from carries oxygen from the lungs or gills to the rest of the body, such as tissues. So hemoglobin actually contains 4 chains, 4 polypeptide chains. Here you can see the 4 pyrrole rings, and in the center you can see the iron. Fe plus 2. So 4 pyron rings is surrounded by the iron ion. So this is generally the structure of the hemoglobin. So in infants and in adults, these hemoglobins have different chains. Like if you talk about the infants, so in infants they contain 2 alpha chains and 2 gamma chains, while in case of adults, normal humans have 2 alpha chain and 2 beta. As the adults grows and infant grows, so the gamma chains are gradually and slowly converted into the beta chains. So the each of them, like hemoglobin, normal adults hemoglobin contain 2 alpha and 2 beta chains. So each of the alpha chains contain 141 amino acids, and each of the 2 beta chains contain 146 amino acids. In total, one hemoglobin contains 574 amino acids. Here you can see also the normal weight of 65 kilodalton. So this is the normal weight of the hemoglobin. It's while here you can see hemoglobin range in RBC, it's 27 to 31 picogram, while the mean volume of RBC is 80 to 100 femtoliter. F stands for femto, and micrometer cube, it's the unit of femtoliter micrometer cube. So it's the mean volume. So in this case, one RBC contains 270 million hemoglobin. So by this you can imagine that how small is the hemoglobin. A single RBC contains 270 million hemoglobin. Now we will study the clinical correlation of the hemoglobin if we have a deficiency or a low count of the red blood cells. So which diseases will occur? So megaplastic anemia. So if the normal size of the red blood cell is greater than the normal, so as a result the RBC will burst, and this condition is called megaplastic anemia. While we will study another, the most common disease like anemia, if we have less number of RBC. So as a result, what will occur? RBC, if we have less number of RBC, so oxygen carrying capacity to the tissues will be decreases, and at the result of which we can feel tired and weakness. So this is the another disease, the most common disease. While we also have another disease like the sickle cell anemia. So in sickle cell anemia, the normal RBC shape and color and size would be changed, like the shape similar to the sickle. So they are a little bit round and clot, at this they will clot, and the normal flow of the blood will be retracted. So at this disease we are called sickle cell anemia. We also have hypochronic. In hypochronic, the normal iron capacity in the hemoglobin will be decreases. So if we have a low concentration of iron in the hemoglobin, so as a result of which we can face the hypochronic condition. While microcytic is the least folic acid in our folic acid. If we eat greater amount of folic acid gradients and like that, so we didn't feel the RBC. We didn't feel like the RBC deficiency. And of course the last one here is the thalassemia, so the most common disease. So next we will study about the leukocytes. It's also called white blood cells. So the normal count in case of men is 500 to 11,000 WC for microliters. Types like the granulocytes and the agranulocytes. So as name indicates, the granulocytes have a granules, and this type of they have a green like appearance, while the agranulocyte didn't have any granules and they didn't have any green like appearance. So granulocyte we further divide into three types like neutrophils, eosinophils and basophils. So neutrophils is generally multinucleated. They work against the bacterial and fungal infection. So it's also occurs as a first response to the microbial and the fungal infection. So neutrophils works in like that way. While eosinophils, if you talk about the eosinophils, so this is a binucleated. You can see here they have two nucleus, and they works against the parasitic infection and allergic reaction like inflammatory reactions. While in case of basophils, so basophils also bi or tri, they have different nucleus, and its most important function is to respond against the allergic and antigen response. By what they will do if we have allergy or like that, so they produce or release the histamine, so histamine causing vasodilation like that and prevent us from the allergic reactions. And the most important thing here, eosinophil also produce the basophil also produce the mast cells. So mast cells are also called mastocytes or labrocytes. It is a resident cells of connective tissues that contains many granules rich in histamine and heparin. So mast cells generally contribute to hemostasis. So they act as a first line of defense against the antigen and entering the body through the skin, to the skin and mucosa. So they prevent us from the allergic response. Next we have agranulocytes. So agranulocytes is generally divided into monocytes and lymphocytes. So monocytes are a kidney-shaped nucleus, and it acts as a phagocytics like mechanism of against the pathogens, and it presents so monocytes generally act like it represents or presents the antigen to the T cells, and T cells will fulfill or final the work of the antigen. And monocytes are small, eventually become the macrophages, and we know that macrophages are responsible for the initiation of the adaptive immune response. So at this stage they become mature. So monocyte we divide into the phagocytosis and the dendritic cells. So phagocytosis we know already that it helps in destroying the pathogen, while in dendritic cells they destroy the antigen by helping the major histocompatibility class 2 this protein. By the help of this protein they will destroy the pathogen. Next we have lymphocytes. So lymphocyte we divide into three types, like the natural killer cells, the T cells, and the T cells. Initially we divided into two types, like natural killer cells and small T cells, but this T cell we can divide into the T killer, T suppressor and T helper cells. So natural killer cell, as name indicates, that this directly kills the microbes without any extra or wasting the time. They directly kill the microbes. While in case of thymus, so we have thymus killer cell, thymus suppressor cell and thymus helper cells. So thymus killer cells here they help by the major histocompatibility protein 1. They activate that protein and kill the microbes. While in case of T suppressor cells, they suppress the activity of the antigen and destroy them. While T helper cell activate the B cells, and after that B cell will destroy. Here we destroy the antigen. Here I want to differentiate the T cells and B cells. Generally T cells are produced in the bone marrow and mature in the thymus, while B cells produce in bone marrow and mature in the bone marrow also. So T cells are migrants, like they mature in the thymus. That's why we are called T cells. So that's all the phenomena of the leukocytes. Next, number third is platelets or thrombocytes. So platelets or thrombocytes are small colorless cells, fragments in our blood that form clots and stop or prevents bleeding. So this is the main cells that prevents us from the bleeding and help in clotting formation. So platelets are made in our bone marrow, like the it's a sponge-like tissue inside our bone. Bone marrow contains stem cells that develop into red blood cells, white blood cells, and platelets. So intrinsic and extrinsic pathway. As name indicated, intrinsic pathway will occur inside the soft tissues, while the extrinsic pathway is the outer lining of the that soft tissues. the outer layer of the soft tissues. So intrinsic pathway start when the at initial stage the factor 12 will be activated. After that they will activate the factor 11, and factor 11 will activate the factor 9, and at this stage the like they both work together. So after that extrinsic pathway will be start. So what they will occur, extrinsic pathway activate the factor 7. So if factor 7 activated, so factor 9 and 7 collectively activate the factor 10. So by this the prothrombin here they will convert into the thrombin, and fibrinogen as we get the thrombin. So after that and the thrombin convert the fibrinogen into fibrin, and they start forming the fiber-like structure. As the fiber-like structure will form, so as a result we receive what the clot will occur. So it's like simple. At initial stage the factor 12 will be activated. After that the factor 11, and 11 will activate in case of intrinsic pathway. So factor 11 will activate the factor 13. So as factor 13 will activate it, so the extrinsic pathway will start, and extrinsic pathway will activate initially the factor 7 and factor 13 from the intrinsic, and factor 7 from the extrinsic. They collectively activate the factor 10. So as the factor 10 is activated, so inside the tissues, the soft tissues, the prothrombin is converted into thrombin, and thrombin convert the fibrinogen into fibrin. So at this we have a clot formation. At this clot formation will be occur. Next, here we can see the thrombolytics. So here two types of drugs will affect. Of course, here you can see the two types of drug, the fraxaparin and Xarelto. So these two activated 2 factors and activated 10 factors, which help in the clot formations. So here you can see the thrombolytics. Here you can see two types of drugs like the streptase and the aminase. Trade name is the streptokinase and NS3-palase. So NS3-palase is a thrombolytic drug. It is also known as the N-acylated plasminogen streptokinase. NS3-palase as modified congener of the streptokinase. While the streptokinase is an extracellular protein extracted from the certain strands of the beta hemolytic streptococcus bacteria we use against the streptococcus bacteria. And the streptokinase and aminase, while trade name is the NS3-pals, act with the plasminogen to produce an activator complex, and that activator complex they convert the plasminogen into the pro-thrombolytic enzyme plasmin. In this slide you can see here the streptase. If you like the mechanism of action of these two drugs is like that. If we take in the streptase and the aminase, so these two drugs work like they activate the plasminogen, and plasminogen convert into the plasmin, and the splitting and the clot formation will be occur. So that's like the mechanism of these two drugs. If we talk about the its doses, like parental dose of the streptase is the 1.5 million is the international units, and the half-life is 18 minutes. While 30 units is the aminase, we can take it as a parental dose. Parental dose which will be taken other than oral. So 90 to 105 minutes is the half-life of this. Here you can see the treatment of anemia. We have low number of RBC in our body. So folic acid, vitamin B12, ferrous sulfate, and ferrous gluconate, and Integra. So here you can see the formula of the ferrous sulfate. So mechanism of these drugs, mechanism of action of these drugs is like that. And these all drugs have iron in their formula. So iron combined with the porphyrin and the globin chains to form hemoglobin. So as hemoglobin will form, so we have more number of hemoglobin, and we know that hemoglobin is the main carrier of the oxygen delivery from the lungs to the tissues, so which is critical for oxygen delivery from the lungs to the other tissues. So iron deficiency causes a cytic anemia due to formation of the small erythrocytes with insufficient hemoglobin. So you can see here the concentration of hemoglobin. In this case, it's a result of a patient that in this case the normal range of the hemoglobin, which is around about 120 to 140 gram per liter, but in result you can see here the 91. So it's lower than normal. So in this we can say that we have a microcytic anemia. And MCV. And the MCV here you can see the mean corpuscular volume or mean erythrocyte volume is less than the normal, like 74.7, and normal range is 82 to 95. Here are some differences between the intrinsic and the extrinsic pathway. In case of intrinsic pathway, so this is self-sufficient and depends on factors from within the blood, and it have more steps, longer cascade, while it is slower but stronger or more efficient. And initial stages, it starts with factor 12 and involves factor 8, 9, 11 and 12, while in case of extrinsic pathways has less steps, shorter cascade, and depends on the outside of the factors. In case of extrinsic, so as name indicated, it's outside from the tissue. So faster but less efficient, and starts with factor 7 and involves factor 7 only. So now we are here to study the folate cycle. So folate cycle generally play a central role in cell metabolism. Among the important functions of folate cycle are the delivery of 1 carbon units to the methionine cycle. So methionine cycle for the use in the methylation reaction, and the synthesis of the pyrimidine and purine. So rule of folate cycle in human body. What is the rule? So folate cycle is a vitamins like B vitamins. It's made as name as here you can see it's made up of vitamin B6 and B12. So that is naturally present in many foods. So your body needs folate cycle to make DNA and other genetic material. It's helping making the, as later you can see in the mechanism of the folate cycle, they're helping forming the DNA and RNA and other genetic material, and also needed for the also they're making the other materials they need for your cells to divide. So a form of folate called the folic acid. So folic acid generally used in the fortified food and the most dietary supplements. So the mechanism of folate cycle is start from the homocysteine. Homocysteine is an amino acid that are produced when the proteins are broken down. So if the level of the homocysteine will be decreases in our body, so this condition we are called the hyperhomocysteinemia. So if we have greater amount of homocysteine, so this can lead us towards the arterial damage and blood clots, and also in our blood vessels. Clots formation occur in our blood vessels. And if we have a greater amount of the homocysteine, so if the level of homocysteine is greater, so it will be indicate a deficiency in the vitamin B12 or the folate. So by this we can indicate that we have a deficiency in vitamin B12 or folate. So the reaction is start like that. Homocysteine, first of all, they in the presence of this enzyme, the 5-methylene tetrahydrofolate. This convert by the addition of a methyl group, this convert the homocysteine into the methionine, while they use some ATPs, and as a result methionine converted into the S-adenosyl methionine, and after that they detach the methylene group which added initially. If the methylene group is detached, so they convert they form here the DNA and RNA, the genetic material which help in transferring the heredity and from parents to offspring. And again they convert into the S-adenyl homocysteine, and the addition of water they again, and the cycle is continuous like that. We already talked about the hyperhomocystinemia. So if we have greater amount of the homocysteine, so as a result of which our blood arterial damage will occur and the blood clots in our blood vessel will be occur. Here you can see the some drugs like folic acid, ferrous sulfate, ferrous gluconate, and ferrous fumarate. So the interior dose of these drugs are folic acid like 200 to 400 and half-life is about 2 hours, while this ferrous sulfate, this is the drug which have both the doses, like we can take it either interior dose or parental doses. So you can see here in the table. So that's all. Thank you. Thanks a lot.